IISER Pune uses nanovesicles to improve cancer drug delivery. Encapsulated drug produced 94% silencing of AURKA, leading to 25% inhibition of RELA by encapsulating the poorly water-soluble anti-cancer drug lysertib in polysaccharide nano-sized balls for vesicles. Researchers at the Indian Institute of Science Education and Research IISER Pune have been able to increase its uptake by breast cancer cells and achieve greater ability to restrict cancer cell growth. Better uptake of the drug when encapsulated meant lower concentration 0.02 microgram of the drug was sufficient to restrict cancer growth significantly better than the free drug. The study, which was started about 6 years ago, gains importance as the drug by targeting the grave accent on AA now selectively inhibits RELA protein, thereby disrupting cancer cells' ability to grow without anchorage-independent growth. Anchorage independence in tumors is what allows cells to spread and grow at new sites in the body. Metastasis. Target protein. Since producing inhibitors against RELA protein is challenging and the existing inhibitor is not specific to target both RELA and RAL, the researchers chose to target aurora kinase AURKA instead. Aberrant AURKA activity has been implicated in cancer formation, and its inhibition could potentiate the effect of chemotherapies. When AURKA is silenced, RELA protein, which is present downstream, gets automatically and selectively inhibited. The researchers embedded breast cancer cells in 3D gels, collagen or 8 rows, and incubated them with nanovesicles containing the drug and a fluorescent dye. In 15 days, the breast cancer cells grew into a colony in the 3D gel. About 75% of the encapsulated drug was retained in the nanovesicle and carried into cancer cells during the 15-day period. While the free drug did not inhibit the colony-forming ability of cancer cells, the number of colonies was significantly reduced in the presence of nanovesicles loaded with the drug, says Professor Navarrat Balasubramanian from the Department of Biology, who led the team along with Professor Mainakam Anand from the Department of Chemistry, IISER Pune. The encapsulated drug produced 94% inhibition of the target AURKA, leading to 25% inhibition of RELA. The drug did not affect RAL. As a result, there was 38% inhibition of anchorage-independent growth of cancer cells. In comparison, at 50%, the RAL inhibitor achieved greater inhibition of RELA than the encapsulated drug, but the inhibition was not specific. RAL was inhibited by 64%. The inhibition of anchorage-independent growth was only 33%. What was interesting is that besides being specific to RELA, the encapsulated drug achieved greater inhibition of anchorage-independent growth than the RAL inhibitor. This despite the RAL inhibitor achieving double the inhibition of RELA than the encapsulated drug, says Professor Balasubramanian. So the lysertib drug did better than the existing RAL inhibitor and led to better inhibition of anchorage-independent growth. That's the most striking observation of the study, he says. The results were published in the journal Molecular Pharmaceutics. For this study, the nanovesicles were encapsulated with only one drug, lysertib, in the hydrophobic envelope, while a fluorescent dye was loaded in the hydrophilic core. But the 2014 study published in the journal Nanoscale by the team showed that loading the nanovesicles with two drugs enhances the therapeutic efficacies against cancer cells. Hydrophobic and hydrophilic anti-cancer drugs loaded in the envelope and core of the nanovesicle respectively performed better than free drugs and synergistically killed breast and colon cancer cells. The proof of concept study was published in 2012 in Biomacromolecules. We are aiming to start animal studies in mice to study how well the drug contained in nanovesicles is able to inhibit breast cancer tumors. We are also using this drug loaded in nanovesicle to inhibit and study a role of AURKA and RELA in normal and cancer cells. So it also becomes a tool to understand a role of these proteins in cells, he says. Fabricating nanovesicles. To make the nanovesicles self-assembled, we undertook some modifications to the dextrin polysaccharide using a molecule from cashew nut shell extract, says Professor Anand. Since both are from bio-based nanosystems, the nanovesicles are not toxic to cells, and possibly human, and are biodegradable. Professor Anand and his team started working on making the nanovesicles self-assemble way back in 2008, and in 2012 they succeeded once they knew the structure that is required to make the nanovesicle with modification. The nanovesicle is bilayered and is held together to form a stable vesicle through hydrophilic-hydrophobic interactions, Professor Jayat Anand says. Since the nanovesicles are about 120 nanometer in size, they are easily taken up by cancer tissue but not normal tissue. Also, nanovesicles with the drugs loaded become water dispersible, thus increasing the uptake by cancer cells. We are working to make the nanovesicles even more specific to cancer cells, Professor Jayat Anand says. The drug-loaded nanovesicles are cleaved by esterase enzyme once inside the cell. In spite of being cleaved, the drug is released slowly in a controlled manner over 8 to 10 hours, Professor Jayat Anand says. This would be important for how nanovesicles act in tumors,